Hello and welcome to your Microbiology Bio 203 video lecture on chemical principles. I'm Mr. Kennedy and I'll be your guide as we explore this topic together. As we enter into the world of chemical principles, the first subject for us to discuss is the basic structure of atoms. Chemistry is the study of interaction between atoms and molecules. The atom is the smallest unit of matter that cannot be subdivided into smaller substances. Atoms interact to form molecules. Atoms are composed of electrons, protons, and neutrons. Electrons are negatively charged particles, protons are positively charged particles, and neutrons are uncharged. Protons and neutrons exist in the nucleus of an atom, whereas electrons orbit it. Atoms with the same number of protons are classified as the same chemical element. Each different chemical element has a different number of protons. The atomic number is the number of protons in the nucleus. The atomic number should be balanced by the number of electrons that are orbiting the nucleus at ground state. These are the elements required for life. Electrons are arranged in electron shells. The electrons orbit the nucleus as they are attracted by its opposite charge. The electron shell that the electron orbits in corresponds to the respective energy that the electron possesses. Atoms form molecules by combining to fill their outermost shells. The number of missing or extra electrons in the outer shell is known as an atom's valence number. Molecules hold together because the valence electrons of the combining atoms form attractive forces called chemical bonds between the atomic nuclei. A compound is a molecule that contains two or more kinds of atoms. Water, H2O, is combined form of two hydrogen atoms and one oxygen atom. Ionic bonds. The number of protons and electrons are equal in an atom. Ions are charged forms that have gained or lost electrons. In order to form an ionic bond, an electron from one atom must be passed or exchanged with another atom. In the example that you see here, sodium has one extra electron in its outer shell, which it will pass to chlorine, which is short one electron in its outer shell. As a result of this, the sodium atom has an outer shell that is now full. However, it also has a positive charge, as it is lost one electron necessary to balance the positive charge of the nucleus fully. Chlorine has gained an extra electron, making its outer shell also full. However, as a result of gaining the extra electron, it now has a negative charge, as it has an electron that cannot be accounted for by the positive charge of the nucleus. The respective atoms with opposite charges are attracted to each other, and thus the ionic bond forms. Cations are atoms that lose electrons and become positively charged ions. Anions are atoms that gain electrons and become negatively charged ions. Ionic bonds are attractions between ions of opposite charges. One atom loses electrons and another gains. Here illustrated again is the connection between sodium and chlorine, forming sodium chloride, or table salt. Covalent bonds form when two atoms share one or more pairs of electrons. Covalent bonds are stronger and more common in organisms than ionic bonds. For example, let's look at hydrogen. Hydrogen is unstable as a result of only having one electron in its outer shell. It can form a single covalent bond with another hydrogen atom to achieve stability, illustrated here. Carbon can form as many as 4 covalent bonds with individual hydrogen atoms, each sharing 1 electron apiece with carbon in order to fill its outer shell and achieve stability. When doing so, carbon can form methane. Hydrogen bonds form when a hydrogen atom that is covalently bonded to an oxygen or nitrogen atom is attracted to another nitrogen or oxygen atom in another molecule. This is an example of how hydrogen bonds can form between water molecules. Molecular weight and moles. The sum of the atomic weights in a molecule is the molecular weight. 1 mole of a substance is its molecular weight in grams. When considering water, H2O, hydrogen has an atomic mass of 1 on the periodic table. In water, there are 2 hydrogens, therefore 2 is multiplied times the atomic mass of 1, which equals 2 AMUs, or Daltons. On the periodic table, oxygen has an atomic mass of 16. There's 1 oxygen in a water molecule, so we simply take 1 times 16, and the atomic weight of oxygen is 16 AMUs, or Daltons. To get the molecular weight of the entire molecule of water, you simply add the hydrogen atoms to the oxygen atoms, and your molecular weight is 18. Chemical reactions. Chemical reactions involve the making or breaking of bonds between atoms. A change in chemical energy occurs during a chemical reaction. If energy is given off, a reaction is exergonic. If energy is taken in or absorbed, the reaction is considered endergonic. Synthesis reactions occur when atoms, ions, or molecules combine to form new, larger molecules. Anabolism is the synthesis of molecules in a cell. Decomposition reactions occur when a molecule is split into smaller molecules, ions, or atoms. Catabolism are decomposition reactions in a cell. Exchange reactions are part synthesis and part decomposition. Atoms or molecules literally change partners or change places in these reactions. Reversibility of chemical reactions. Some chemical reactions can readily go in either direction. Each direction may need special conditions. If a reaction is reversible, a double arrow is indicated in the center of the reaction. Important biological molecules. Organic compounds always contain carbon, hydrogen, and typically are structurally complex. Inorganic compounds typically lack carbon, are usually small, and structurally simple. Inorganic compounds include water. Water is inorganic, however, it is polar, which means it is charged. It has an unequal distribution of charges. It is considered the universal solvent as it will interact with and or bond with any other polar substance and undergo dissociation, forming solutes. Water can also, as a result of its hydrogen bonds, absorb heat and can behave as a temperature buffer. This is an illustration of the unequal distribution of charges in water and how water can form hydrogen bonds. Not only will these hydrogen bonds allow water to absorb heat it also gives water its other unique properties of surface tension, adhesion, and cohesion. Acids, bases, and salts. Substances that dissociate into one or more hydrogen ions or protons and one or more negative ions can form acids, bases, or salts. This is an example of an acid. Hydrochloric acid can dissociate into a positively charged proton and a free chlorine. Substances that dissociate into one or more hydroxide ions typically form bases. Sodium hydroxide is an example of a base. Here you see sodium hydroxide dissociating into a free sodium ion and a hydroxide ion. Substances that dissociate into cations and anions, neither of which is hydrogen or hydroxide, make salts. Sodium chloride is an excellent example of a salt, since it is the actual molecule that makes up table salt. When it dissociates, we get a free sodium and a free chlorine. This is sodium chloride, or table salt, dissociating. The concentration of hydrogen ions in solution is expressed as pH. pH is equal to the negative log base 10 of the hydrogen ion concentration in solution. Increasing hydrogen ion increases acidity. Increasing hydroxide ion increases alkalinity. Most organisms grow best between pH 6.5 and 8.5. This is the universally agreed upon pH scale. Organic compounds. Structure and chemistry. Organic compounds commonly contain hydrogen, oxygen, and/or nitrogen in addition to carbon. The chain of carbon atoms in an organic molecule is the carbon skeleton. Functional groups bond to carbon skeletons and are responsible for most of the chemical properties of a particular organic compound. This is an example of just a few organic compounds: methanol, ethanol, and isopropanol. This table shows a variety of functional groups that can attach to and work within organic compounds. This is a second table of functional groups for organic compounds. Structure and chemistry. Identify the functional groups in an amino acid. In blue, we have the amino group, and in salmon, we have the carboxyl group. Small organic molecules can combine into large macromolecules in the world of organic chemistry. Small organic molecules are referred to as monomers. As they combine into large macromolecules, they're referred to as polymers. Monomers are joined by dehydration synthesis or condensation reactions. As the name implies, a bond is formed as water is removed between two complex molecules. The opposite is true when the bonds are broken. Water will be simply added and will have what is referred to as a hydrolysis reaction. This is a rudimentary illustration of dehydration synthesis. Carbohydrates. Carbohydrates serve as cell structures and cellular energy sources. They include sugars and starches and consist of carbon, hydrogen, and oxygen with the formula CH2O to the N. Many carbohydrates are isomers, molecules with the same chemical formula but different structures. Monosaccharides are simple sugars with 3 to 7 carbons. Glucose and deoxyribose are examples of common monosaccharides. Disaccharides are formed when two monosaccharides are joined in a dehydration synthesis reaction. Disaccharides can be broken down by hydrolysis, which again is the addition of water to break the bond. This is an example of glucose and fructose undergoing dehydration synthesis when read from left to right. When read from right to left, a hydrolysis reaction occurs. Polysaccharides consist of tens or hundreds of monosaccharides joined through dehydration synthesis. Starch, glycogen, dextran, and cellulose are polymers of glucose that differ in their bonding and function. Lipids. Primary components of cell membranes are lipids. Lipids consist of carbon, hydrogen, and oxygen, are nonpolar and insoluble in water. Simple lipids are referred to as fats or triglycerides, which contain glycerol and fatty acids formed by dehydration synthesis. This is an example of a simplistic lipid with a glycerol and a single fatty acid chain attached to it. If this was a true triglyceride, there would be 3 fatty acid chains attached, not just one. Simple lipids can also be referred to as saturated and unsaturated fat. Saturated fat has no double bonds in the fatty acid. It is saturated with hydrogen. Unsaturated fat has 1 or more double bonds in the fatty acids. Unsaturated fats come in cis and trans varieties. In the cis variety, the hydrogen atoms are on the same side of the double bond, and in the trans, the hydrogen atoms are on the opposite side of the double bond. This is a more accurate representation of a full triglyceride, a glycerol attached to 3 fatty acid chains. The chains that are straight are saturated, and the chain that is bent is unsaturated. Complex lipids. Complex lipids contain carbon, hydrogen, oxygen, phosphorus, nitrogen, and or sulfur. Cell membranes are made of complex lipids called phospholipids. Glycerol, 2 fatty acids, and a phosphate group are needed to make a phospholipid. Phospholipids have polar as well as nonpolar regions, and this is how they appear. The polar region is attracted to water, whereas the nonpolar region is not. When put into an aqueous solution, the polar region will seek water, and the tails or fatty acid tails will simply seek each other. Hence the formation of a lipid bilayer, which makes all life on Earth possible. Steroids. 4 carbon rings with an OH group attached to one of the rings is the common structure of a steroid. Steroids can be a part of membranes that keep membranes fluid. This is a picture of cholesterol, which is a steroid that helps maintain the fluidity of the common cell membrane. Proteins. Proteins are made of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Proteins are essential in cell structure and function. Proteins are enzymes that speed up chemical reactions. Proteins can also be transporter molecules that move chemicals across cell membranes. Proteins can act as flagella that aid in movement, and some form bacterial toxins and other cellular structures. The primary building block of a protein is an amino acid. Amino acids contain an alpha carbon that has an attached carboxyl group and amino group and a side group. This is the basic architecture of an amino acid, which you've seen from an earlier section of this video. Amino acids have 20 different side groups that make each amino acid unique. Aside from the core architecture of an amino acid, which is identical, the side groups give each amino acid its unique properties. Amino acids will exist in either of 2 stereoisomers, the D or L form. The L form are most often found in nature. Peptide bonds are the bonds that are responsible for holding amino acids together. They are formed by dehydration synthesis and broken by hydrolysis. This is an illustration of the formation of a peptide bond. If you will imagine the arrow going the opposite direction, then you would have hydrolysis. Once an amino acid chain is formed, we have the primary structure of a protein. The primary structure of a protein is little more than a long sequence of amino acids referred to as a polypeptide chain. The secondary structure of a protein occurs when an amino acid chain starts to fold and coil in a helix or pleated sheet arrangement. The tertiary structure occurs when the helix or sheet folds irregularly, forming disulfide bridges, hydrogen bonds, and ionic bonds between amino acids in the chain. The quaternary structure of protein consists of two or more polypeptides interacting and/or binding with one another. In this illustration, you can see the full progression from primary structure through quaternary structure. A final note on proteins is that proteins can also undergo denaturization. Proteins prefer specific temperatures and pH levels and will lose their shapes if those temperature and pH levels are violated. Conjugated proteins consist of amino acids and other organic molecules. Glycoproteins, nucleoproteins, and lipoproteins can form when proteins interact with carbohydrates, nucleic acids, or fats. Nucleic acids. Nucleic acids consist of nucleotides. A nucleotide is made up of a 5-carbon pentose sugar, a phosphate group, and a nitrogen-containing purine or pyrimidine base. Nucleosides are made up of just the pentose sugar and the nitrogen-containing base. DNA is the most well-known nucleic acid. It contains deoxyribose, exists as a double helix, and has 4 bases: adenine, which hydrogen bonds with thymine, cytosine, which hydrogen bonds with guanine. The order of the nitrogen-containing bases forms the genetic instructions of the organism. And this is a picture of DNA. RNA is a close cousin to DNA. RNA stands for ribonucleic acid. RNA contains ribose, which is a single-stranded molecule, and has the 4 bases adenine, which hydrogen bonds with uracil, cytosine, which hydrogen bonds with guanine. There are several kinds of RNA that play specific roles in protein synthesis, which will be discussed at a later date. This is a picture of uracil nucleotide in RNA. Another nucleotide is adenosine triphosphate, or ATP. Adenosine triphosphate is made of ribose, adenine, and 3 phosphate groups. It is the primary energy transport molecule in all living things. This is what ATP looks like at a molecular level. ATP stores the chemical energy released by some chemical reactions, can transport it through cells or the body, and release that energy when phosphate groups are pulled apart by hydrolysis. This is an illustration of that event. And this concludes our coverage of chemistry for Microbiology Bio 203.